Hello viewers, in this video we are going to discuss about classification of viruses. On the basis of shared properties, viruses are grouped at different hierarchical levels of order, family, subfamily, genus and species. International Committee for Taxonomy of Viruses have described almost 2000 virus species placed in 3 orders, 73 families, 9 subfamilies and 287 genera. This data is as per the 8th meeting of ICTV, that is International Committee for Taxonomy of Viruses. Different hierarchical levels like order, family, subfamily and genus, their names end in particular words, or we can say some particular suffixes are used at the end, right? So for in an order name, the order name will end with the virales, right? Like for example, mononigevirales. Okay, so virales, whenever there is a viral word, means it is an order. Okay, so group of this virales is a group of related viruses having negative strand RNA genome, right? And family name ends in viridae, right? So this mononigevirales have 4 families, including this paramyxoviridae. So viridae suffix is used for a family name. Okay, viral family name. Paramyxo is a Greek word that means by the side of, and myxo means mucus, by the side of mucus. So paramyxoviridae contains 2 subfamilies, including paramyxovirinae. So virinae is a suffix for subfamily. So this family has 6 genera, including rubella virus and morbivirus, right? So this rubella virus has a type species. This genus has type species mumps virus, so MUV, and this morbivirus has a type species, that is measles virus, MEV, right? There is a system of virus classification known as Baltimore System of Virus Classification. Here, viruses are classified based on mechanism of genome replication and mRNA synthesis. So how their genome is getting replicated and how mRNA is synthesized. Depending on that, viruses have been grouped in seven groups, right? So 1 through 7. Now you will see that first group is of the viruses having double-stranded DNA genome. Second is having single-stranded DNA genome, and third group is having double-stranded RNA genome, right? And likewise, fourth is having plus strand RNA genome, and fifth is having negative strand RNA genome. Sixth is having single-stranded RNA genome, and seventh is having double-stranded grabbed DNA genome. Now, if you carefully see, third, fourth, and fifth, these are having RNA genome as well as sixth also. So from third group to sixth group we have RNA genomes, double stranded RNA genome, plus strand RNA genome, negative strand RNA genome, and single strand RNA genome. So viruses have either DNA or RNA as the genetic material. So now here in group 3, double stranded RNA genome is there. And you will say that plus strand RNA genome is actually a single stranded RNA genome, and negative strand RNA genome is also a single strand RNA genome. Then why a separate group is made for single stranded RNA genome? Right. So we'll shortly see that how their mechanism are differing. Group 1 viruses, which are having double strand DNA genome, they replicate through genome replication. Double stranded DNA gets replicated into double stranded DNA directly. And mRNA synthesis, a general process, double stranded DNA to mRNA transcription, right? So this is really simple. Then single stranded DNA genome, single stranded DNA replicates through double stranded DNA copy. So ssDNA to dsDNA, then again dsDNA to ssDNA. And mRNA synthesis, again there is no RNA intermediate, single stranded DNA to double stranded DNA, and then double stranded DNA to mRNA. So first single stranded DNA genome will always be converted into double stranded DNA genome, then followed by either copying into single stranded DNA or replication transcription into mRNA. Now double-stranded RNA genome. You see here all the RNA species are there. RNA, RNA, RNA. And for replication, double-stranded RNA gets converts into single-stranded RNA, and then it gets copied as replicated as double-stranded RNA. So no DNA intermediate here. mRNA synthesis, double-stranded RNA to mRNA synthesis directly. And plus strand RNA genome, first plus strand RNA is copied into minus strand RNA that acts as template and then give rise to plus strand RNA. And likewise, mRNA, because it's a plus strand RNA genome, so itself is a mRNA, right? So, and negative strand RNA, minus strand RNA is first converted into plus strand RNA, which converts finally to minus strand RNA. No DNA intermediate, no DNA intermediate in third, fourth, and fifth group. mRNA synthesis, minus RNA, minus strand RNA get copied into plus strand mRNA, that is mRNA. Now that we have specifically dedicated a particular whole group to single stranded RNA genome having viruses, so replication occurs through a double DNA copy, as well as mRNA synthesis occurs through a DNA copy. So that's the difference between these single stranded RNA genome and this separate group, right? So there is a DNA intermediate for replication as well as mRNA synthesis. Now, double-stranded gapped DNA genome viruses replicate through a double-strand DNA intermediate. Gapped DNA first get copied into a double-stranded DNA, then it gets transcribed into plus-strand RNA, and then it gets you know, copied into a minus strand DNA, and then minus strand DNA serves as a template for gap DNA, double-stranded DNA synthesis. So there's a long process. First, dsDNA is converted into DS, gap dsDNA will be converted into dsDNA, then plus strand RNA, then minus strand DNA, then gap dsDNA. Then mRNA synthesis occurs through a double-stranded DNA copy. Now, first gapped double-stranded DNA converts into double-stranded DNA, and then that get copied, transcribed into mRNA. So this way, this Baltimore system of virus classification is complete here. So I hope you understood that how these 7 groups are classified depending on their genome replication and mRNA synthesis, and also how the single-stranded RNA genome viruses are kept as a separate group because they replicate and synthesize their mRNA through a double-stranded DNA copy. Okay, so thank you guys for coming to this channel. Keep coming and find new videos. Do subscribe to this channel. Thank you.